Good morning, and thank you for allowing me to come and talk with you a little bit about some of the work we've done in this area. You heard Vandana previously talk about the importance of phenotyping, and what I'm going to talk with you today is a little bit about how we've used artificial intelligence and facial analysis to help further that effort. So, like Vandana, I'm also a clinical geneticist, and often what we do is try to make diagnoses using both phenotypic data and genetic data, but also thinking more broadly about other more common diseases. We know that the environment plays a major role, and I'm particularly interested in teratogens and how they have the ability to impact the developing embryo and fetus in causing an ultimate phenotype. So obviously these are three legs of a stool that we have to understand. One of the biggest advances that's been talked about already is in the genetic analysis. And about 10 years ago, there have been a lot of advancements made in the ability to get good genomic data that many of you may have seen this diagram already, but we haven't been able to advance similarly in other areas such as phenotyping. So one of the first efforts to try and get at this problem of getting a really robust phenotype was led by a group of geneticists a few years ago to try and standardize the nomenclature. And they worked together and came up with the elements of morphology that you can now see hosted on the NIH website. In our field, in dysmorphology, we focus on the face, hands, and feet because that's the most variable regions of the body. And not surprisingly, they came up with over 400 features that one could annotate going through that. Once we standardize the phenotype, the next question becomes, how do we capture it? And I was part of a study site for the National Children's Study a number of years ago when we realized we were building this research platform with a great amount of environmental data, a great amount of genetic data, but the question was, were we capturing the entire phenotype so that we could really look at the entire clinical picture? And so we came up with this protocol that involved 15 photographs and 3 videos, 8 of the head and neck region, and 4 of the hands, and 3 of the feet, along with the accompanying videos, to try and catch as much of those 400 plus phenotypic features as possible. We then did a pilot study and had those images analyzed by a panel of geneticists to try and see if they could annotate the features reliably off of the photographs. And it worked, but as you can imagine, it was a very time-consuming process, taking about an hour for each geneticist to go through one individual. But this was a poster that I took to a conference talking about this assessment tool that we had developed, and the organizers, because of their prescience, were smart enough to put my poster next to another poster that was talking about the use of computer-aided facial recognition in a particular diagnosis. So as we had figured out how we could capture enough imaging to analyze a phenotype, we hadn't quite figured out how we were going to automate that process in order to get a significant amount of information on a number of individuals, whereas our colleagues had been working on that exact thing. So we began a collaboration from this point forward, looking at how we could incorporate the image capture and analysis. So the software, it's called Face2Gene, and it's developed by a company called FDNA. And the way the software works is an image is inputted into the system, and the system identifies a certain number of fiducial points on the face. Then ratios are calculated between those points, and the face is converted into a numerical description. This numerical description of the face is then compared against approximately 300 known syndromes where they have pretty good numerical data about, and then the system outputs the top 20 conditions that most closely resemble that of the patient. From the user side, in the clinic, this is what the app looks like. So here we have the image of a patient, and the system provides the list of possible conditions underneath. And then the provider can add additional details. So for example, the image isn't going to capture growth parameters or the presence of heart defects or renal anomalies. So the user can then input that to further refine the differential. And then finally, working through the differential, the user can then compare their patient with the average face for that particular syndrome, and a heat map is presented so that one can see what areas of the face are matching. So colors that are hotter, reds and oranges, are showing where there's a higher degree of match, whereas those that are cooler are showing lower degrees of match. So as I mentioned, I'm particularly interested in teratogens, and one in particular is alcohol. And so I just want to give you a little bit of background on fetal alcohol syndrome so that you can understand the nature of the project that we did from here. So FAS was first defined in the late 60s, early 70s as a particular pattern of malformation seen in women who consumed alcohol during pregnancy. And the features were pretty consistent. They involved growth deficiency that involved length and or weight, but interestingly persisted after birth. And they had a characteristic face that you might be able to appreciate from the training doll that's shown above, as well as the patient below. But namely, the openings of the eyes, what we call the palpebral fissures, are relatively short, so you don't see much of the white or sclera of the eye. And then the upper lip is noted to be quite thin. And the philtrum, which has a typical 2 columns that extend down from the nostrils to the upper lip, along with the groove in between, is indistinct. Finally, we know that alcohol has an effect on the developing forebrain, and the result is neurocognitive deficits, particularly in the areas of higher executive function. When FAS was first described, the full phenotype is what was initially recognized, but it didn't take long for folks to realize that there were milder effects of alcohol, and so the term fetal alcohol effects was used to characterize anything that wasn't the full FAS. Subsequently, they've been recategorized as fetal alcohol spectrum disorders, with these 4 diagnostic categories that come from an Institute of Medicine report about 20 years ago. And these categories actually still stand, although we've refined the criteria for making each individual diagnosis. So we have full FAS, which requires all of those features I talked about earlier, but there's also partial FAS, where you have some but not all of the physical findings. And then 2 other categories, alcohol-related neurodevelopmental disorder and alcohol-related birth defects. Alcohol-related birth defects really are just a series of anomalies that we know are more common to present with individuals who've been exposed to alcohol. For example, we know the heart, the kidney, the vertebra are more sensitive to effects from alcohol. But alcohol-related neurodevelopmental disorder has been a bit of an issue to diagnose because there are no outward features of this particular condition. The diagnostic criteria require, obviously, the presence of alcohol exposure, but also a very characteristic pattern of neurobehavioral impairment. And this requires a pretty detailed neuropsych evaluation, and therefore we can't typically make this diagnosis in children under the age of 3. Again, to have the resources to conduct that kind of assessment has been a bit of a challenge, and so ARND remains quite difficult for most clinicians to make. It's also probably the more prevalent form of FAS that we see in this country, and therefore it's really imperative that we find ways to get at that problem. So we decided to attempt the facial recognition software to see if it could discriminate between the different types of FAS. We had 36 subjects with full FAS, 31 with partial FAS, 22 subjects with ARND, and then 50 controls that were matched based on race, ethnicity, and age. And the images that we had were the frontal view for these kids, as well as a 3/4 view demonstrated in the bottom right. And that's really to look at the philtrum a little bit closer. The way the study was done is we took those images, we split them randomly into 2 groups, 50-50. The first group was used to train the system so that it would learn to recognize, using artificial intelligence, what was FAS, partial FAS, ARND, and control. And then the remaining set was used as the test set, and the system would try to categorize based on what it had learned. We repeated this using 10 rounds, and then compared the rate of discrimination between the manual assessment of the clinician against the computer-aided assessment. So this is a receiver operating characteristic curve, which essentially plots sensitivity against 1 minus specificity. So as you might imagine, a perfect test with 100% sensitivity and specificity would be a very steep curve on the front end and then would flatline at 1. And you can see for any FASD, when we compare the manual score in red to the computer-aided score in blue, they both performed reasonably well. But what was really interesting was when we looked at the individual categories, in the case of ARND, the computer-aided score actually outperformed the manual score. And I can show you the tabulated data just so you can see that, where in any FASD, the manual score and computer-aided score are very similar, and we saw the same thing for FAS and partial FAS. But in the case of ARND, the area under the curve for computer-aided score was 0.84, whereas for the manual score it was at 0.74. And these were pretty impressive differences. So we then said, well, what is it that the computer is picking up that we are not picking up? Because we don't have any physical characteristics in our clinical criteria. And what we learned from the artificial intelligence is that ARND looks different than FAS and partial FAS. So on the right, it's showing us in red the areas that it was getting cues from for FAS and partial FAS that are within the areas of the midface that we know alcohol impacts and in the regions around the eyes. But for ARND, the artificial intelligence was actually picking up cues from all over the face. And so there must be something that's more broad but is really a subclinical from a provider standpoint to be able to detect. So what we learned about this is that facial analysis software with artificial intelligence may be able to help further delineate phenotypes at a subclinical level, and that it may even, if we think about other environmental exposures, instead of waiting for some major birth defect to arise, perhaps a system like this could pick up even more subtle changes from different things that are in the environment impacting embryonic and fetal development. And so there may also be some external physical biomarkers for conditions like FAS and other potential exposures that this could be used to detect. And then finally, in the field of genetics, if you've ever had to refer to a geneticist, you know the wait times are quite extensive. And a software like this may be able to be deployed in a primary care setting, perhaps as an initial screen, to get patients to a diagnosis in a much more rapid and accurate way. With that, I'll conclude my talk and hand it back over.